Okay, let's go through 7.2 quickly. So 7.2, we're talking all about the regulation of enzyme activity. So it's kind of a continuation of what we talked about in 7.1. It's actually in the same chapter of the book. So it's all just the same thing, except this goes into a lot more detail on allosteric regulation and on covalent regulation. So we're going to start with allosteric regulation. And the one that it talks about most is ATCase. And it talks about ATCase because ATCase is a really good example of feedback inhibition. So when you have a lot of CTP in the cell, it's basically a signal that you have a lot of nucleic acid biosynthesis. And so what will happen is that as it becomes very saturated, that CTP will then self-inhibit the creation of CTP. And then ATP actually increases the creation of CTP. So the way that I think about feedback inhibition is if I'm like an elf in Santa's workshop and I'm making like little Barbie dream castles, and then at some point I make so many of them that they start hitting me on the head and then I can no longer create more Barbie dream castles. That's like what I think about. It's self-inhibiting. So feedback regulation happens a lot with our hormones. So if you create a lot of a certain kind of hormone, it will eventually negatively feed back to what's going on. And so you create less of that hormone. End of story. So when we talk about this type of allosteric effector, another type of allosteric effector that you can remember is 2,3-BPG. So you remember that allosterically affects hemoglobin and changes the conformation of hemoglobin, which then affects other subunits and other components. So that is very similar to allosteric regulation with ATCase. So with ATCase, when you add ATP, you're stimulating ATCase. When you add CTP, you're inhibiting ATCase. And so it shows you a little bit of a diagram of this initial V versus aspartate. And so what it's going is it's saying basically you have a lower Km when you're stimulating ATCase and you have an increased Km when you are inhibiting ATCase. So that's really all that's about. So for your T state to R state, your actual product is your N-carbamoyl L-aspartate. It's not necessarily the most important thing to remember. I think the biggest thing to remember is that ATCase produces CTP. It's an example of feedback inhibition. That CTP and ATP relationship is very self-regulating. You create more CTP. CTP becomes very saturated. It ends up having the enzyme produce less. And I say enzyme loosely, but yes, enzyme less. So ATCase is now producing less ATC. And then with more ATP, the enzyme's going to be producing more CTP. So it kind of is in this kind of big loop of stuff. And all of that is nucleic acid biosynthesis is the whole kind of point of it. And so yeah, and it's allosteric. So don't think active site, it's allosteric regulation. So that also means it is reversible, sorry. It is reversible, allosteric, reversible. But we're going to get into some that are irreversible as we go. So now we're going to talk about phosphorylation. So phosphorylation is going to be covalent. So phosphorylation is right here on the board when you kind of look at it. So phosphorylation is really common with your serine, your threonine, and your tyrosine. So that's going to add a negative charge to the enzyme through the addition of an inorganic phosphate. And what this does is it changes the conformation and the molecular structure overall. And so that is going to affect the regulation of the activity. So the example that it gives you is glycogen phosphorylase. So glycogen phosphorylase is a dimer. A lot of things are different, like it'll give you, oh, this is a trimer, this is a dimer, just kind of things to think about and have in the back of your mind, but not as necessarily like important to understanding the concept. So the biggest thing to remember with this example of phosphorylation is that in this example, you have a kinase, which phosphorylates, and you have a phosphatase, which dephosphorylates. And why is this important? It's important because you have different enzymes causing the phosphorylation or the dephosphorylation. And so in something like, as we're going to get to adenylation, you actually have the same enzyme both upregulating and downregulating, depending on certain components that we'll get to. In this, you have different enzymes. Kinase phosphorylates, phosphatase dephosphorylates. It takes that inorganic phosphate off. So you're going from your T state to your R state. It's really common in all of them, obviously, because you're either producing or you're not producing. And then for glycogen phosphorylase, the kind of interesting thing is that glycogen phosphorylase, it degrades glycogen. So that's the whole idea of glycogen phosphorylase. And so glycogen is a storage of glucose in liver and muscle cells, and glycogen, or in the storage of glucose in glycogen. You need to generate glucose for energy, so you need to access those glycogen storage, and you need to be able to get access to that glucose, which is why when you have glucagon and epinephrine, you're going to that R state because you want your glycogen phosphorylase to start breaking down that glycogen and giving your body glucose. So if a tiger is coming at you, you better hope that epinephrine is allowing you to activate your glycogen phosphorylase so you have sugar, because you want that sugar to go to your cells to allow you to run away really fast. But then insulin is going to be shifting it to your T state and stabilizing that T state, because now your body's producing insulin. It's like, okay, like we need to chill out. We need to start kind of letting the cells take in some of this glucose and use it to make ATP and like chill out a little bit on our levels of glucose in the blood. You don't want high elevated levels of glucose constantly. That's diabetes. So don't want to do that. So then we want to talk about a little bit proteolysis. So this is really common in digestion. It's also really common in blood clotting. And proteolysis is an example of something that is irreversible. So it's not that the rest of them are not, because generally covalent modification, we think of irreversible, but with proteolysis specifically, the lecture wanted to remember this specifically is irreversible. So in proteolysis, what you have is this technically happens in a lot of different things, but in the example, it wants you to remember pepsin. So pepsin is what is responsible for the part of what is responsible for the breakdown of food. So it breaks down food by catalyzing the hydrolysis of peptide bonds. You'll see it here. And so basically you can break this down into 4 different steps that are going to happen. So in your nutshell, you have pepsinogen, acid, cleavage, active site. So what these mean: pepsinogen, step 1, you have inactive pepsinogen. And inactive pepsinogen, you have an active site blocked by 44 amino acid N-terminal fragment, which is a lot to say. That just pepsinogen is a zymogen. It is inactive. It can't do anything yet. It's a precursor to the actual active thing that you want to be generating product, right? So then the next thing that's going to happen is you have acid. So the acidic environment in your stomach is going to cause a conformational change. That conformational change is going to result in the cleavage of the internal segment. So now this thing blocking the active site is getting cleaved, and then the active site is going to be exposed. So your nutshell: pepsinogen, acid, cleavage, active site. So that active site is exposed, and now things can bind, regulate, and create different kinds of products. Cool. So a little bit extra on ATCase, because we kind of went back and forth on ATCase a few times in the lecture. So your ATCase is also going to be called C3R3. And the reason that it's called C3R3 is because you have 3 ATCase dimers, and each of those dimers are going to have 1 catalytic subunit and 1 regulatory subunit. One additional component of this is to actually have active ATCase, you need 2 of those 3 ATCase dimers popped together. So that means you're actually going to have C6R6, because you need 2 to have active ATCase. So you might see that, especially if you go through the book. A lot of times it calls it C6R6. Cool. And again, think of C3R3, C6R6, catalytic, 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 regulatory, 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 because you have 1 per 3 of the ATCase dimers. Cool, cool. All right, so then going into more covalent modification. So this seems very scary, the adenylation and the uridylation, but it's not that bad actually. So if you really just kind of break it down into the bare bones of what you probably need to know, hopefully, I didn't write the test. So adenylation. So when you're talking about adenylation, you're talking about the same enzyme, both adenylating, and deadenylating. And so you're shifting from R to T using the same enzyme. So that's different if you'll remember phosphorylation, where you have kinase and phosphatase. You have 2 different enzymes. Really easy to remember. You see that phosphatase, you're like, that's taking that phosphate off. You see that kinase, you're like, that's adding that phosphate in. But now you have the exact same enzyme. It's glutamine synthetase adenylyltransferase. So if you see it, you don't necessarily know, is that adenylating, or deadenylating? You just don't know yet. So the way that you are going to figure out, is it adenylating or deadenylating, is uridylation. So they're actually combined. Don't think of them as separate things going on. It all happens in one general mechanism. So when you're thinking of glutamine synthetase and you're thinking of kind of what is going on in this entire thing, it's actually about E. coli, which is like, we're talking about bacteria. That's okay. So the example is with E. coli. So this example of the adenylation and the uridylation is specific to E. coli and glutamine synthetase in E. coli, because glutamine synthetase is an integral in bacterial nitrogen metabolism. So it's super important for the E. coli because they need to metabolize nitrogen to be able to continue living their life. So this is the way in which they're going to do that. So a little bit of easy talk here for what are these things. So uridylation is literally just the adding of the UMP covalently. Important. Adenylation is just adding AMP covalently. So uridylation, add a UMP covalently. Adenylation, add AMP covalently, and that's it. That's all you gotta know. So when is it going to happen? So when you have high ATP and high metabolite alpha ketoglutarate, which all of this is happening down here, you have these kind of like blue things going on. You can look this up in the lecture as well, or I'll just send you a picture of this. So you're gonna have high ATP or high metabolite alpha ketoglutarate. And so what this is going to do is that it is going to cause the uridylation activity. So this is adding a UMP. So you see you go from this little pink guy, does not have a UMP, and then it's going to add that UMP in the presence of these two components of ATP and alpha ketoglutarate. So now that is happening. But what is happening when we talk about AMP, when we go this direction? We are getting rid of the AMP, which means that we are deadenylating because you're getting rid of the AMP. So you are uridylating because you're adding a UMP, but then you're deadenylating because you're taking the AMP away. And a good way to remember this is that when you're adding the UMP and you're taking away the AMP, you're creating an active enzyme, right? It's in the R state now. It's happy in the R state. So this guy's happier than this guy. So this AMP, when it's added, is basically stabilizing the T state, the inactive state. And so to get to the R state, you got to take that AMP away, which means you're deadenylating, and just know that they're basically opposites. If you're adenylating, you're deuridylating. If you're deadenylating, you are uridylating. So you're adding UMP, taking an AMP away. You're adding an AMP, you're taking UMP away. So they just act kind of reverse of each other. So that means that conversely, your inorganic phosphate and your glutamine, when those are super high, this is going to cause the deuridylating activity. So now I'm taking that UMP away. And when I take that UMP away, I am then going to cause adenylating activity. I'm going to add the AMP. So breaking this down into like more chemistry, you can think of, you have an alcohol group over here, and that's going to be adenylating. And if you have a UMP group, you're deadenylating. But to me, I think it's a lot more simple just to think of it as kind of a flow. ATP and alpha ketoglutarate, those are giving you lots of energy. Energy is great. You want energy to be able to take that AMP away because you want to have activity. So if I want activity, I need ATP. So cool. I have ATP, I've got alpha ketoglutarate. I'm going to basically activate this enzyme, and to activate that enzyme, I need to uridylate and deadenylate. Now, conversely, if I have high glutamine, I have high inorganic phosphate, I don't have ATP, I don't want activity, I want to stop the activity. I'm like, let's chill out. So if I want to chill out and bring him back to here, I need to first deadenylate and then adenylate. So I'm going to take that UMP off, I'm going to stick an AMP on. So that's really the simplest way to kind of go through it. So you have a little bit of allosteric and covalent modification when you're talking about this, because when you're talking about these guys down here, which are going to be either the reason that you're uridylating or deuridylating, because you have your ATP, your alpha ketoglutarate, your interphosphate, your glutamine. Well, those things are allosteric effectors, right? So they're not permanently binding, but they are affecting that component in order to affect the uridylation, which then affects that AMP and that adenylation. So not too hard, but not too easy either. But if you just try to break it down for yourself into more simple terms of going forward, going back, energy, no energy, and just kind of thinking through it in a more logical way, to me, it sticks in a little bit better. So there you go. That is both the lecture and the book 7.2.